So, good afternoon, everyone. So, my controversial topic I'm going to talk about today, I want to really pose this question. Are stable isotope analyses key in resolving dietary disputes and niche partitioning in the fossil record? And to explore this question and to hopefully answer it, I'm going to be looking at the Messel lagerstätte, which is this famous lagerstätte in Germany that you might have heard of. So the aim of this presentation is to evaluate the paleoecological applications and effectiveness of stable isotope analyses. And the Messel lagerstätte presents a perfect case study to test this hypothesis, and it has actually been explored by previous authors. So the presentation is going to consist of two parts, and first part I will dive into what approaches have been used in the past to reconstruct the food webs in Messel. So how do we know the relationship between the individual taxa? And the second half of the presentation will dive into the debate about the large so-called pteroduck gastonis, which is found at this site, and what its role in the ecology and its diet was. So to introduce the Messel fossil locality, for those of you who don't know of it, it's a world-class lagerstätte, which means a site of exceptional preservation, and it preserves an early Eocene ecosystem that was about during a greenhouse climate, and it's located in Germany, some 20 kilometers southeast of Frankfurt. The formation of the Messel site is actually quite an interesting one from a geological point. It preserves a Marl Lake environment, which actually formed when localized volcanic activity and igneous intrusion, that's molten rock, came into contact with groundwater, and that caused a lot of steam and generated a huge explosion, which created this volcanic pipe and crater, which subsequently filled in and became a freshwater lake. The question of why so many fossil taxa are so exquisitely preserved at Messel is still a subject of current debate, and this is something we can return to in the questions, but it's likely due to, in part, anoxic conditions at the bottom of the lake. So how about reconstructing the Messel food webs? So as I just said, the Messel site is exquisite as it preserves soft tissue of the animals that were alive at the time, which is quite incredible. And you can actually see that here in this fossil snake, which is quite nice that we're going back to fossil snakes for a moment there. We can see the skeleton, but we can also see there's this like dark shadow following around it, and that is original organic tissue that's been preserved. And I think this being a vertebrate fossil, this has to be probably one of my favorite vertebrate fossils, because we have inside this fossil snake a fossil lizard, and inside the fossil lizard we have a fossil beetle. So what this one fossil is actually showing is a potential trophic structure that the beetles were fed on by lizards and lizards were fed on by snakes at Messel, which I think is incredible that we have this preserved. And this is what some authors have actually suggested. However, we need to be careful because these rarer isolated discoveries risk to oversimplify the food webs. We know that this snake, for example, fed on lizards, and these lizards fed on at least some beetles, but we don't know if the snake or the lizard, for example, fed on other types of food sources. The high levels of fossil preservation at Messel have also led some authors to investigate more abundant types of fossil evidence, and they are less glamorous. They are coprolites. So a study done in 2001 by two authors examined over 150 fish coprolites from the ancient Messel Lake, and actually revealed a previously undocumented and diverse invertebrate fauna. And we can actually see within this coprolite all these small fragments of insects and invertebrates. And from this, they were able to construct a simple food web. And the advantages of doing this is that we know that this is based on direct consumption. It's not just a faunal list that has been tiered with the smallest animals at the bottom and the biggest at the top. And also, in part, preserves this complexity of the Messel Lake, which was before then wasn't preserved. The large vertebrates are, but the smaller invertebrates were not. But there are disadvantages. The fish coprolites only preserve the lower trophic levels and not the higher ones, which And also there is talk about it being unquantitative. How can we quantify this with other sites and other parts of the locality? The difficulty, for example, is also shown by it's only really showing small fish, what small fish ate. Everything above that is kind of left to speculation. And the particular, I've highlighted here, a particular genus of fish, which we'll return to, which is, in this diagram has been placed as a fourth level consumer. But how can we be so certain about that? And also, to make matters worse, we have unintentional digestion. So these fish were swimming in the Messel Lake, and there was algal blooms, and unintentionally they were swallowing these algae. So it also just complicates the entire picture more. So what can isotopes do in helping us understand this a lot better? So a study 6 years later re-evaluated this earlier 2001 study through the scope of carbon-13 and nitrogen-15 stable isotope analysis, importantly on that original organic soft tissue that you saw on the snake just previously. So here, the carbon-13 has been used as a proxy to distinguish between terrestrial and aquatic food sources. So if you look at this diagram in the top left-hand corner, as we get more negative carbon-13, we're more likely to be aquatic food sources, and as we get more positive carbon-13, it's more likely to be terrestrial. Nitrogen-15 isotopes have been used as a proxy to indicate the trophic position. So things at the base of the food web should have a low nitrogen reading, and as you progress up the trophic level towards the top of the food web, we should have higher nitrogen values. And the advantages of this is that actually these methods have actually been used in modern lake environments, and the results are quite similar. It largely confirms these earlier unproven studies which looked at the coprolites. And actually, the fish, for example, has been now thought to be more of an opportunistic feeder rather than a full-fledged carnivore. Also, this particular diagram can distinguish between terrestrial and aquatic food webs. So, for instance, the carbon-13 measurements within insects that were analysed overlap with carbon-13 measurements in the plants, confirming that insects indeed fed on plants. But interestingly, some insects like the giant ants had the nitrogen-15 values higher than other insects, suggesting carnivory. So there was these giant carnivorous ants at Messel, which is quite interesting. We also see, again, increased nitrogen-15 values in taxa such as crocodiles and other larger invertebrates. But there are disadvantages with this technique, potentially diagenesis and other sorts of environmental factors that can influence the signal. And it seems that this can't be used on its own. We need to use this alongside other direct and indirect paleontological evidence to really draw some firm conclusions on the structure of the food webs. And also, this was a first of its kind study, but a larger sample sizes would be needed to do in the future. So quickly moving on to part 2, what was the diet and ecology of this giant terra duck, as it's called, Gastonis? So this Gastonis species has actually been discovered at Messel from a single isolated femur back in 1965. But it was originally described from early Paleogene of France in the early 1850s. And the notorious American paleontologist Edward Drinker Cope actually described a similar animal, Diatryma, from New Mexico 20 years later. But we now consider that Gastonis and Diatryma are actually considered the same taxon. So this was a big animal, standing at about 1.8 meters tall. The Gastonis would have dwarfed all other animals within its ecosystem in the early Eocene. And this has led some potential authors to question the diet of Gastonis. Potentially, was this a huge hypercarnivore that was preying on our early mammalian ancestors? Or perhaps is it as simple as that? And although a nutcracking or herbivorous lifestyle has been considered, Whitmer and Rose in 1991 suggest that Gastonis was more likely to be a predator, specialising potentially in bone crushing or scavenging, on the basis of the adaptations of the skull morphology. Its skull was so large and reinforced, these authors just couldn't see why it would be a a herbivore. This had to be a carnivore. And this interpretation had lasted. And you might remember the BBC Walking with Beasts trilogy back in the 90s. And the popular media now portray these large birds as active carnivores preying on these like small horses. But how accurate is this interpretation? So Whitmer and Rose did do a biochemical analysis on Gastonus, and it showed that it did have a very powerful bite. However, a more recent morphofunctional study on the jaws of Gastonus have suggested that the position, the muscle scars on the skull, which you can see here, would correlate with, which are called MAME, correlate, the size of these muscles actually correlate with herbivory. So carnivorous birds, modern day birds, have small muscles, whereas larger birds that you break nuts, for example, have larger muscles. And this is something we see in Gastonus. So potentially, could it have been a herbivore? So how do we prove this? So again, we come back to isotopes, and the same study that suggested this also employed, again, carbon-13 stable isotope analysis on Gastonus bone appetite, which was then compared to measurements taken on carnivorous and herbivorous birds from the modern day. This time, carbon-13 was a proxy for diet, so decreasing carbon-13 would relate to increasing herbivory. And the advantages of this is that the Gastonus material was sampled from a multitude of different French localities, so this could limit if an overall signature was produced, this would suggest maybe a more original signal rather than a diagenetic one. And this is the actual the graph here, and we can see a difference between the values if it was a carnivore and compared to the values in the shaded area of if it was a herbivore. And another advantage is it's using the biomechanical study alongside isotopes to draw more conclusive results. The measurements of the carbon-13 isotopes here disprove a carnivorous lifestyle, as the authors suggest the readings could only be produced, these carnivorous sorts of readings could only be produced if the prey that Gastonus ate exclusively ate C4 type vegetation, which had not evolved yet in the early Eocene. So this has led some authors to suggest it is a herbivore, and again, diagenesis could potentially be a problem. So overall, these C3 results have more recently been backed up with recent calcium isotope data, which is another trophic tracer, and kind of support this hypothesis that Gastonus was in fact a herbivore. So maybe we should see less of this and more of this. So just to quickly conclude and summarize my presentation, stable isotope analyses present a new and modern approach of reconstructing the structure of fossil ecosystems. However, the main conclusion from this presentation should be that stable isotopes should always accompany other contemporary approaches when constructing past food webs and diets. And we have shown this, that the combination of both stable isotopes with other sorts of paleontological information can produce more accurate results. For example, disproving the carnivory theory and potentially supporting existing interpretations in fossil food webs. And that is me. So I'll stop sharing my screen, and I'm happy to take any questions.